Our next speaker is one of the co-organizers. It's Morten Scheibye Knudsen from the Center for Healthy Aging.
Thank you, Daniela. The slides are coming through? Good. So thank you very much for the invitation. I really appreciate the organizers, great organization here. So my talk is entitled Targeting DNA Repair for Aging Interventions. And so let me just reload this. And so my lab tries to understand, basically try to connect the phenotypes of aging with molecular changes that we see in our cells. So we all know what happens with aging, I think. You know, we develop graying of hair, facial wrinkles, we get redistribution of subcutaneous fat, for example. You get pigmentation changes in your skin, and then you're done. So that's a very depressing start to a talk, I guess. But a lot of other stuff also happens. And so here we sort of try to look at the combined prevalence of features that occur in a person or in a population of elderly as they get old. So graying of hair is the most prevalent feature. About 98% of everybody will develop graying of hair. Other features are less prominent but more clinically relevant. For example, cancer. About 40% of people viewing this stream, unfortunately, will develop some sort of cancer. This is unfortunately the numbers as we have them. Dementia is between 10 and 20% of everyone will develop some sort of dementia. So there's a huge range of features that are associated with aging. And what drives this phenotypical variability, we don't really understand. We can define aging as a time-dependent increase in risk of death. And this is this is the result of, or this sort of corroborates this statement. So this is the 1-year risk of death as a function of your age. This is data from the Social Security Administration in the US. And so as you can see, as we get older and hit about 60, 70, you get a markedly increased risk of death. And if you look at this as a survival curve, so this is the surviving fraction of a population of people as a function of age. And this is actually the Danish population. You can see that there will be a mean age, of course, around 80. But this data is, I think, more interesting in the context that we actually know we can combine survival data with, for example, pharmacy data. So each line here is a cohort of people taking a prescription drug. This is a huge, a massive data set, more than 1 billion entries. And we can then see that certain medications are associated with longevity and certain medications are associated with shorter lifespan. And part of this, one of the first ones we looked at was, of course, metformin. And we do see this slight lifespan extension effect of metformin, but the survival curve actually crosses after a few years. So I think this is supportive of metformin because these people have diabetes. So this, I think, is very interesting. This is data we're still investigating. Aging, of course, occurs in many different organisms. So we study humans, mice, and fruit flies. And of course, the main difference between us and a mouse is our genomes. And so there are even certain organisms that don't really seem to age. This is an animal called a hydra that has the same constant risk of dying as a 19-year-old human. So the point is that aging is not really, it's not magic, it's a biological process and it's influenced by our genes. And we all remember this quote from Lord of the Rings. Of course, another piece of evidence for this is that you can get mutations in single genes that give premature aging. And these genes are, many of them are involved in DNA maintenance. This is often caused by defects in DNA repair. So DNA repair is important in aging. I think we've heard that this morning. So what if we could stimulate DNA repair? Could that be a way to reduce aging, or the rate of aging and the consequences of aging? And so to do this, we turned to our favorite philosopher, Nietzsche. What does not kill me makes me stronger. So if you're a cell, and if you get stressed, and if you don't die, you come out on the other end actually having increased resistance to the stressor. So this is called a Hemetic response. So if you irradiate cells with some sort of irradiation, for example, X-rays, at a low dose, the cell survives. They are able to cope with a higher dose next time. So our idea is to exploit this response to stimulate cells to upregulate their DNA repair responses. And this is done also in collaboration with in silico medicine. And this is the basic overview. We use neural networks on a large amount of training data to characterize or stratify about 20,000 compounds and then find compounds that could stimulate DNA repair. And so we screened 15,000 compounds. And we found a number of hits that could potentially stimulate DNA repair. So how do we screen them? This is in collaboration with Peter Sikora and Amelia Technologies. So we use the Comet chip, where you can directly measure DNA damage in the form of strand breaks, and we could then test if our compounds could do something about strand breaks. And we also wanted compounds that elicit or that gives the Hemetic response, but does not induce DNA damage. So we only want the beneficial response, but not the detrimental damage. So this is how the data comes out. You have different concentration doses here, and then you have different compounds here. And if you quantify it, you can see for compound A, you get an increase in level of DNA damage as a function of concentrations. At very high doses, you have very few cells, so this is maybe a little bit misleading, but I think it's quite obvious that within the increasing dose and increasing time, you get more DNA damage. So this compound we do not want. We want a compound that does not induce DNA damage. And in fact, many of the top hits do not seem to induce DNA damage. So that was good. So we go on and look closer at these compounds. We screened them then for whether or not they would activate the DNA damage response. And some of them did. This is done with high content microscopy. You get more 53BP1 and gamma H2AX, which are markers of DNA damage. You get more of these markers in the cells. But many of them don't really do that. So some of them even have sort of this biased activation. They activate gamma H2AX and decrease 53BP1. Compound C is a quite interesting compound that we're exploring more. I'm not going to talk more about it. And then there are other compounds that seem to be interesting. So are they actually able to protect cells from radiation-induced damage? And so quite strikingly, if we give cells a very, very high dose of ionizing radiation, which will kill typically 90% of the population of cells, there are certain group of molecules that completely immunizes the cells to this very high DNA damage dose. So these are very interesting, of course. One of them, drug F, actually decreased the amount of senescent cells when we tested them in culture by looking at beta-gal staining, and also increased lamin B1, which decreases with senescence, and so drug F seems to increase this. And indeed, if you look at replication induced senescent, we can see that drug F actually increases the ability of cells to grow in culture. And this is an experiment that, of course, takes a long time. And I think Garrick, this is Garrick from the lab, he really loved this experiment and would love to repeat it many times. He's in the audience, so you can't see it, but he's laughing. So how else can we measure senescence? So one big issue, and I think people in the senescence field, which is not a field that I'm part of, would agree that it's very difficult to have sort of one single marker for senescence. We tried to do this by developing a neural network that can predict senescence based on DAPI staining, and this is done by Indra Heckenbach, who's most often, he's a PhD student in my group, but is actually staying at the Buck Institute. And so we used a neural network to segment images to recognize nuclei, and then we can train a neural network to detect beta-gal staining, and then nuclei that have beta-gal staining close to them we then label as being senescent. So if we do that, we get a very, very high degree of accuracy in our neural network's ability to predict senescence based on beta-gal. We have also done it without beta-gal, just simply by ionizing radiation or replicative senescence. And we get typically around 90% accuracy, so it's really extremely accurate based solely on the nucleus size and shape and whatever else is in the DAPI staining. And so using this approach, we can see that there's actually fewer senescent cells when you treat with drug F. So it seems to be reversing senescence in this way also. And in fact, if we look at what happens to the cell, they actually appear to, not only does senescence appear to be reversed, they actually start expressing pCNA again, and there's cell growth compared to control. So they're going back in cycle and starting to grow again. How does it work? Well, one way it works is that it appears to be able to reduce the amount of DNA damage in replicative senescent cells. So this is quantified here. And also, if you treat cells with ionizing radiation, wait for 7 days to induce, for senescence to be induced, and then treat with this drug F, it actually reduces the amount of persistent DNA damage foci. So these are foci that are thought to not being able to be repaired, and this compound actually appears to be able to reduce the amount of these foci, which is, I think, quite remarkable. So what do they do in animals? To do this, we turn to Michael Peter, another PhD student in the lab, who is starting up a company called Track Bio, where we can actually measure these things in live animals. Here we're using flies, but we can also do it in mice. And so we've trained a neural network to be able to recognize flies and track them over time. And that means that we can actually count the flies, figure out how their motor function is and their behavioral interactions are. And so using this approach, we get data like this. So this is the movement pattern of flies at a certain time point when they're young. You can see flies are happily walking around. When they're drug F, they're also happily walking around. An old fly here seems to be walking around less, and for some reason it's more walking horizontal and vertically. I'm not sure why that is, but an old fly in drug F is really still very happy and is walking around quite a lot. And this is the quantification then, and you can see with increasing age, you see a decline in the mean distance traveled by the flies, and this is not the case with drug F. And this is, I think we heard that yesterday, that we know that walking distance and walking speed is an extremely good predictor of health with aging, both in humans and in animal models. And we also see this actually in flies, and this is reversed by drug F, so this is quite interesting. And drug F also appears to be able to increase the mean lifespan of flies. So this is really very exciting, and we are exploring further with this drug. I would also like to point out that we're doing the same thing with other types of DNA damaging agents, UV radiation, oxidative stress, where we are finding some quite interesting results also. I don't have time to go into that. And so finally, I just want to highlight that we are starting a new journal called Frontiers in Aging. It's part of the Frontiers family, and it will open up in a few weeks, and I highly encourage you to contribute to the journal. If you are interested in running a research topic, you can also do that. This would be really great to get your support on this. And with that, I'm actually going to stop here and take any questions. This is my group, by the way, and I would like to thank my funders and collaborators.
Okay, thank you very much. We have a few questions on Slack. We can start with one question from Marco De Maria. 7 days post-IR cells might be irreversibly locked, whatever you do to them. Do the levels of CDK inhibitors change?
That's very interesting. I don't think we've actually looked at that, but I mean, we do see that they actually start growing again even after 7 days when we treat them with this compound. So they do come back somehow. But I don't think we've looked at CDKs. Garrick is shaking his head. He's saying no.
Okay, one more question about drug F. Wilbert Vermeij is asking, have you tested drug F in a DNA repair deficiency mouse model?
We've tested it in cell lines. We're testing it in a fly model. We know, so the drug F, we actually know there is a putative target of drug F that we know, and it makes it not particularly compatible with testing in mice. So we actually have a number of modifications to the compounds that we've tested, and also found some other compounds that work. So we will test it in mice. We're not there yet. We've tested in progeroid cell lines, in attaxic telangiectasia cells, and Hodgkin-Gilford progeria cells, and Cockayne syndrome cells. And in all these cases, it decreases the amount of gamma-H2AX53BB1 staining. Garrick is nodding his head. So.
Okay, and one final question from Alexander Merseburg. What is the data source for the survival curves of individuals on different drugs?
Yes, so this is a great question, and this is where it's good to be in Denmark, because in Denmark we have for about 40 or 50 years been monitoring everybody's drug use, and we have very good registries of survival, and so we can combine these registries relatively easily. So we have basically we know what everybody's taking of prescription drugs. We know what everybody is when everybody is born and when they die, and we actually also have their medical history. And then it's basically looking at which people are taking what drugs and how does that affect lifespan. And so I think another interesting point here is that we can also look at combination of drugs. So we can start looking at whether or not people that have been treated with metformin and statins, how does that impact lifespan, and so forth and so forth. So this is registry data that's very unique to Denmark. And it's, as I mentioned, more than 1 billion prescriptions and covering these like 5 million people.
Okay, thank you very much.